Good evening, everybody. See, today's topic that has been given is very, narrow down, and of course, it is very good that they have. I mean, people have given me this topic to highlight. It's a pleasure for me in this platform that I can share my experience with a live example where we have set up the laboratory commercialized plant tissue culture center. Of course, it was started in Parlakimudi campus also, and thereafter it was also started in Andhra Pradesh campus of Centurion University. And this is I just wanted to show the audience the complete setup that we have. And this center we have is capacity of more than 5 lakhs plants. Actually, it is 10 lakh plants. And at present, we are doing 5 lakh plants. And with this experience of setting up and establishing the center, I would like to take the audience through this particular plant tissue culture and its applications in agriculture and allied sciences. Here, I will not be taking up the class like students. It will be completely like a commercialization setup where we need a whole set is different from what we teach in the classroom. So to go further, this is one, whatever the pictures that I am showing completely without taking even a single picture from Google. This is complete activities that we are taking up, and this why we have to do a plant tissue culture and how it can be a part of agriculture is one criteria that we have to look into, where usually plant tissue culture, when we talk about it may people may talk about genetic engineering and modifying the complete crop as such, but the other side, when you look into it, it is only growing of certain plants in vitro condition. Many of you, if students are there, then I'll just get into a small terms, meanings, and other things. In vitro is growing the plants inside the glass tubes and in the laboratory, where in vitro culturing we do and then grow the plants in high number. And one is the objective is to grow a high number of plants in vitro condition. The second would be high yielding plants will be taken up and then will be propagated further. And the other thing is each should be genetically feasible. That means genetic fidelity will be taken care, and it will be produced based on the principle of totipotency. In such environment, we require. Am I audible? Hello. Yes. Okay. These are the very important criteria that we need to maintain. One is committed leaders or committed staff. Not like academic sector. Of course, academic, I'm not seeing it down. But then when commercialization comes, it becomes a more important criteria where committed leaders and staff is very important, and they should know, and we should train them very well in this particular sector. And the second thing is a very systematic and timely execution of the work is important. Even the shift and shift-wise work also can be taken care, and it will be systematically executed. And the application oriented research and production is very important. Along with the commercialization, it is not that just simply commercialization is important. It is that research wise and production wise, it should be very systematic, and it should be taken care where along with maintaining sterilization and hygienic condition in the complete center, inside as well as outside the premises. So this becomes very important to take care of all these criteria uh, to produce a high number or large number of plants or propagates. So in this case, one is I'm actually taking up into dividing this presentation into two. The production is by micropropagation, another is production by vegetative propagation. This micropropagation is involved, which involves the complete internal laboratory is concerned, and we have to prepare large number of large liters of media, nutrient media, and produce a large number of plantlets. So this you all know that if we have to concentrate on micropropagation, then we have to go through several steps. One is we have to select the explant of banana. At present, we are growing banana, so I'm taking the example of banana. Of course, there are other crops also that we are working: bamboo, gerbera, and chrysanthemum. Of course, that is not at large scale, but banana is at large scale, and this mother plant would be, taken from a field where large bunches of the banana will be produced, and we will do this field survey and then get into the, considering the mother plant. So this explant selection is very important. Once that is selected, the initiation would take place once the nutrient media is prepared with a specific concentration having vitamins, amino acids, macro elements, and plant growth regulators, etc. I'm not getting into the media preparation and other things here because we have a very short time, and I need to cover up how exactly the plant tissue culture is its application part in agriculture. So once the initiation of this explant is done in the media, there is multiple subculturing that we need to do. Multiple subculturing is usually a banana. Usually, when you plant the plant in the field, from then it will take at least 12 months to give the harvest. So in that case, even in vitro condition, when we subculture, when we initiate the propagation, this actually takes 7 months inside the laboratory and 2-3 months in the polyhouse condition, which is called acclimatization, and primary secondary hardening will take place. And this is the overall picture of what exactly is done in plant tissue culture. And these are the, again, live pictures of our center, where 500, of course, now we have taken a large number of inoculants. That means explants, 5,000 inoculants. And from there, we started with a small setup. And then now we are into 5 lakh plants. And mostly in this center, of course, there are local people are involved who have been trained very well for about 3 months, and then we will pay them with a minimum. During training period, we will train them, and then that means we are giving employment to the local people. And then we also employ our ALP students who engage in this agriculture. And from PKD campus, we 3 months to 4 months, and then they will be they'll be absorbed. If they are interested, they'll be absorbed into the center with a good payment. And these are the process that is carried out. There are two important steps: shooting and rooting. Shooting is usually in the explants will be cut and subcultured for several number of times. That is about 6 cycles. Every cycle is about a month. And then the 7th month, we looking at the growth of the shoot, we will transfer the shoots into media which contains a rooting hormone, IBA and NAA, for the stabilization. And Further, these are the activities that carried out for hardening. Once the rooting, you can see the roots that are getting propagated in the media, which contains activated charcoal, and also the rooting media. We call it as rooting media, which will have rooting hormone also, and then transferring that in vitro propagated plant lines into the pluck trays, and then later on into the nursery bags when it grows to a minimum of 15 centimeter. And you can see the left hand side corner, there are n number of plants that are propagating well in the under the polyhouse condition. These are all the pictures that has been taken in our center. So this, I have to highlight here, why I put this picture here is to just to show the audience or the listeners here that we prepare the racks of our own. Of course, Centurion University provides it has industry connected academia concept where there is a lot of workshop activities that go on, and as per our requirement, the workshop, the mechanical engineers, they prepare these racks, and also the laminar airflow chamber. I think I'll just go back a little bit. Here you can see the picture of laminar airflow chamber, which has been prepared, manufactured in our workshop in our own in-house built. And likewise, this rack is also, and also some of the students that are involved, engineering students are involved in making of this racks and laminar airflow chambers. In this way, we are actually, going towards the concept where everything of whatever we use in-house should be made in-house only. And so you can see the explant that what we are using will be the harvest like this, which will have a very big banana, first of all, and very large numbers in the entire bunch. In one hand, you can see around 100, around 30, 40 banana you can see here. And of course, we are expecting the same thing. And this is actually we are expecting in our own field in Andhra University, in AP campus, where we actually are maintaining 1 acre of this complete tissue culture raised banana plants field, and we are going to expect this kind of a bunch. As we are doing this in the first of course, it is a second season, and we are going to expect the harvest in the probably November, December 2023. And this is one of the leaflet which I wanted to show you here is Centurion University is associated strongly with Gram Tarang, which is a company. Because when we have to sell the plants, it should be first thing is DBT certified, and that we are going to get it in a very short time, in another 2-3 months. And we are getting associated. We already have associated with Gram Tarang, and which is called GT plant tissue culture. And here you can see the very important benefits of the complete product, where there is a farmers who are the selling these at a reasonable price. Of course, in the market it is around 20 rupees, and we are actually selling at 10 rupees, and sometimes of course, if they are affordable, then we go for 12 and 15 rupees. And we have a sophisticated laboratory facilities also, where biotechnology and this tissue culture go hand in hand. And we also have a DST project, which is highly funded. And this plant tissue culture products is high yielding. You can see the pictures here. and this is disease-free, especially the virus-free plants are supplied, and best quality plants in all throughout taken care. So all the other details for the farmer who buys the plants are given here. How to prepare the land, what kind of soil it should be, and what is the season that needs to be when taken care to plant this, and what is the spacing that we usually follow up, and then fertilizer, irrigation, and intercultivation practices, is it affordable or not, and how the flowering takes place, and then harvesting. Everything is given here, and the contact details is also given at the end. So this is another picture where our agriculture students are involved and transferring the in vitro plants into ex vitro. These are the plug trays, and after they grow, we actually separate them into A, B, and C grade. And once that is done, we transfer that into nursery plants. So the other part of the just to bring to your notice, where we also propagate vegetatively, because around different kinds, around 200 kinds of ornamental plants we are actually planning, and in this. Some of the where we actually go for flowering plant and ornamental plant, etc. And in addition to the vegetative propagation, we also go for the products, like standardizing the protocol for different crops. Sorry. At present, actually, we have standardized the protocol for chrysanthemum, and also, of course, I have not put the slide for this bamboo. Bamboo also is one of the crop that we have worked on and standardized the protocol. And if anybody is interested, they can come and get the training out of it, and get exposed to such a sophisticated facility, where around a radius of 300 kilometers, we do not find such a center, and most of the things are completely application part. It is not that in the classroom we study about selection of the explant, and then how to maintain or establish the culture media, or how to multiply. We call it as just multiplication, and then go for rooting in different primary, secondary, hardening, etc. It is mostly that you learn about the application part. So that is why I have been stressing upon this. The application part, whatever we study in the classroom, we should be able to bring it to the field, and bring it to the farmers' advantage, where they take advantage of this kind of facility. Of course, farmers need not come and learn the technology, but then whatever the product that we are producing from the technology is definitely they will be one of the best beneficiaries. So in addition to the farmers, there are many employees who are working in our university, are also, I mean, whenever they see the plants growing, just when you enter, if anybody comes into the AP campus, you can see the left hand side of your entry, just in the entrance. Then you can see around 500 plants are growing, and very well they're growing. And when harvest stage will come, then definitely it will be really good looking. And many of our employees do take these plants, and they are maintaining in their own land. So of course, we are actually waiting for this banana to come to a seventh cycle of its growth stage, where once that gets stabilized, then we can propagate this chrysanthemum and bamboo at a higher at a large scale. So and why we are doing that is, like, it should not get cross contaminated, and then whatever the growth that is that we are seeing, you can see here. I'll just show you the last this one. So this setup that we have, and it is already all the bottles are with these kind of plants. Each one explant will give you around 20 plants. Just imagine in vitro that based on this tutor-potency, how the explant is giving a large number of plants whereas in the field at the same time only a single explant will give you a single plant. So that is the advantage, one advantage, where we get from one sucker we will get around 20 plants. Of course, we divide that into A, B, and C grade. Of course, A is a top one, which grows very well, healthy, and then followed by B grade, which is also growing very well, but it is a medium size, and C grade is a poor, or it is not poor. It is called, but it is a lesser than A and B kind of plants. So that is what I just wanted to show. This particular flower, by using this each petal we will get this kind of plantlets. So like that, the bamboo also, we use the buds of the bamboo and then prepare the propagales. Of course, we faced many challenges during standardization of the this protocol while propagating. Sterilization of explant is a very crucial step. Without that, it will not get into the next step. And once the contamination gets entered, the entire process, and we lose a lot of chemicals and the manpower, efforts, and then energy, everything we lose. In this process, we have succeeded in standardizing the protocol for chrysanthemum. And our faculty members are also involved in this. Dr. Anita, Dr. Triveni, myself, we sit. Of course, they are in Lucknow campus, and I'm in IP campus, and we work together and collaborate inter-campus, and then we have come up with these products. And in addition, I just want to say that this center not only works for plant tissue culture and vegetative propagation, it also works for genome-wide analysis, particularly in the field crops like rice, finger millet, and wheat. These are the three crops that we usually work on. This is just for the information, for the gathering, which is here. To get to know about our center, and also where we do not just stick on to the commercialization. Of course, we also work on the research concept, where we have such kind of facilities also. The management is very kind enough to extend the facilities that we have asked for. And then in addition to all that, we have got a CRV, that is DST project, and also trying for this delignification for these tissue culture raised banana plants. Of course, there are not only banana, of course, the DBT will facilitate if once we apply. We are in the process, and it will be applied in this month. So I'll stop here. If I go on, and then there is a lot of other things that we can discuss over, and in the question session, probably we can interact further.